So today we are going to look at TB, tuberculosis. Shall start with doing the classification. The classification shall start with the domain. We start with the domain, followed by the kingdom, here followed by the phylum, the class, the order, the family, the genus, and then the species. The domain is the bacteria. The kingdom is the bacillus. The phylum is the Actinomycetota. The class is the Actinomycetes. Then the order is the Mycobacterium. Then the family is the Mycobacteriaceae. Then the genus is for Mycobacterium. Mycobacterium. Then the species is the Mycobacterium tuberculosis. Tuberculosis. There are so many here. There is Mycobacterium reprae, Mycobacterium, but we are specifically specifying in the Mycobacterium tuberculosis. Let us look at the Mycobacterium tuberculosis. Tuberculosis. When you look at the Mycobacterium tuberculosis, this is myco. Myco means something which is like a fungal. So it has a cellular wall which is in that form, it is in the fungal form, the cellular wall. So that cellular wall has mycolic acid in it. So this myco means fung-like. So our bacteria has this fung-like thing. So in it, this thing is the one which has the mycolic acid, and it is what makes it so difficult to be stained with the gram staining. Actually, then bacteria, it's a bacteria, that's why we include this bacteria. It is bacterium. So the bacterium which has a mycolic thing. This tuberculosis, it causes the disease called tuberculosis. That's why we say Mycobacterium tuberculosis. It causes tuberculosis. Why do we call it tuberculosis? Now, this bacteria called Mycobacterium tuberculosis, when it accesses your lungs, if it reaches the lungs, it causes that some tubercles, some lesions. It makes some wounds in the lungs, and as they try to heal what like that, there are some wounds or lesions, so it leaves, eventually it leaves some things, some lesions. These lesions, they are the ones we are referring as the tubercles. eh? Tubercles. So hence the name tuberculosis. That is our introduction. Let us look at the second part. What are we going to look at? Now, it was 1882. A man called Robert Koch discovered this disease called tuberculosis. And since then, they started calling it Koch's bacilli. They call it, so another name for Mycobacterium tuberculosis, or another name for TB, it is Koch's bacilli. Mycobacterium tuberculosis is an airborne disease. So it is got when somebody is coughing and coughs those bacilli into the air. Or somebody can be singing. So you get it through inhalation. It's got through inhalation of the bacteria saturating in the air. So it is got by inhalation of the droplets that saturate the bacteria in the air. So somebody can be coughing, singing, even quarreling, even laughing, and spits those things in the air. So as a result, you inhale and get Mycobacterium tuberculosis, hence the disease. Now, after getting it, what are the signs to see that you have got the disease? Signs and symptoms. The sign which you see that somebody has got TB, you have to cough for 3 or more weeks. Coughing for 3 or more weeks. When somebody coughs for 3 or more weeks, there you suspect Mycobacterium tuberculosis. You take for the testing. Another thing, second, this is chest pain. Another one is coughing blood. Sometimes you may cough mucus. Night sweats, you might be having night sweats. There is loss of appetite. There is loss of weight. There is also body weakness. Or fatigue. We have also chills. Then we have also fever as the signs for TB. So those are the major signs for TB. Characteristics of Mycobacterium tuberculosis. The characteristics of Mycobacterium tuberculosis. Yeah, characteristic number one. This one, it is a rod-shaped bacilli. 2, it requires oxygen. It requires oxygen to grow. 3, it has a slow growth tendency. It grows slow. It can take 18 to 24 hours to grow. Unlike other bacteria like the Escherichia coli. The Escherichia coli, for it, within 20 minutes, it has already divided. It has a thick cell wall with mycolic acid. So it is too small that it can dodge weak disinfectants. Diagnosis. It is weakly gram positive. It is weakly gram positive. So we usually don't use gram staining. In our previous video, we started about gram staining. So if you don't know gram staining, you can go back to you can scroll until you reach to the video of gram staining and study about it. So it is weakly gram positive, so that's why we usually don't use this gram staining technique to discover it or to diagnose it. We instead use other tests. Majorly here we usually use fluorescent stains like uramine, or we use acid fast stains. Acid fast stains like the ZDN stains or Zell-Nelson staining. We also use PCR. We use the Gene Expert. Majorly here, that's what we use, because I want to teach what is majorly practical. Let us look at the ZDN staining. The Zell-Nelson staining. we start by knowing the principle now. Now, the principle, the key thing here is that the Mycobacterium tuberculosis, has the cell wall. Mycobacterium tuberculosis cell wall has mycolic acid, as we said earlier, which makes it waxy and impermeable to weak stains. Therefore, we apply a strong stain called strong carbofucsin. We apply strong carbofucsin. We apply strong carbofucsin stain with a strong mordant phenol. So this waxy impermeable cell wall, it is hard to penetrate. So we use a strong stain, which is a strong carbofucsin, 1%, with a strong mordant called phenol. This strong mordant phenol helps the carbofucsin to be fixed in the cell wall of the microorganism we are looking for. So, since it is hard to penetrate this waxy part, we apply heat. Since it is hard to penetrate, we apply heat. So this heat will melt. This heat is to melt the waxy mycolic acid and makes it easy for the stain to penetrate through that waxy thing. Once it melts, it will now becomes easy for the carbofucsin to enter inside the cell wall of the Mycobacterium tuberculosis. So once it is there, on cooling of this waxy on cooling of this waxy cell wall, the carbofucsin stain is trapped. On cooling of this waxy cell wall, the carbofucsin is now trapped inside the cell wall. So after it has been trapped, you decolorize. So the Mycobacterium tuberculosis will resist decolorization. Will resist the decolorization of the sulfuric acid. It will resist this decolorization of the sulfuric acid. This is acid alcohol. To resist this, but those ones which are not Mycobacterium tuberculosis or not from the Mycobacterium family, they will be decolorized. Will be decolorized and they lose the color and take up the counter stain, which is methylene blue. So that is the principle which we follow. The wax cell wall is impermeable to the weak. The wax cell wall is impermeable to the weak stains. So we apply the use of strong carboxyl with a strong mordant phenol and apply heat to melt the cell wall. In the process, it will be easy for the penetration of the red carboxyl to penetrate and be received very well inside the cell walls of the microorganism. And on cooling, it will be trapped inside there. So when we counter stain, before counter staining, but when we decolorize, you find that those ones which are Mycobacterium tuberculosis or even Mycobacterium repre, it will resist the decolorization and will remain red. But those ones which are not in the family of the Mycobacterium tuberculosis or Mycobacterium repre, those ones, they will accept the decolorization, will be decolorized, and later when we counter stain with methylene blue, they will take up the blue color of the methylene blue. So that is the principle. Let us see the steps we follow.
Zell, now the Zell Nelson technique. The Zell Nelson technique, it was developed by a man called Zell and later modified by Nelson. Hence the name Zell Nelson staining, which we brief as ZN staining. Now the technique. Which technique do we use? Now the requirements. We need the strong carboxyl. Strong carboxyl. Then we need the running water. We need running water. We need also the acid alcohol, the sulfuric acid. We need also the counter stain methylene blue. Then we need also a clean microscopic slide. Then the staining rack, the drying rack, like that. Steps. Step number 1, get a clean slide and make the smear. Make a smear. Make a smear on a clean slide. That is the first step. Make a smear on a clean slide. This is our slide. This is our slide. Then make a smear. First put here, then first put it like that, then spread it to make a good smear. Very thin. A good smear. After making a smear like that, air dry. After air drying, heat fix. After heat fixing, you now apply strong carboxyl on the staining rack. Apply over flood. Now flood. Flood it with strong carboxyl. Flood it with strong carboxyl. The whole of it, you flood it like this. The whole of it, flood it with strong carboxyl. And let it stay there for 5 minutes. You now apply heat. We usually say with a blue flame. Heat the slide. Heat the slide now. You apply heat. Until steam comes out. So after flooding for 5 minutes, what you do, you apply heat. It is this heat which will melt the wax, and this stain will be now able to penetrate through and enter inside the cell wall. So the sixth part is that you decolorize. With sulfuric acid. Which is acid alcohol. This one is acid alcohol. The one we use. There's a mixture of alcohol in this acid with that acid. Then, after decolorizing with the acid alcohol, this decolorization, it takes like 1 to 2 minutes. So after the decolonization, within the 1 to 2 minutes, the next part is to wash off the acid alcohol. After washing off the acid alcohol, the next part now here is to apply the counter stain. You counter stain with our methylene blue. Methylene blue. So after that, you wash with clean running water. After washing with the clean running water, here our 10th step, after washing with the clean running water, you now do the air dry. Air dry. After air drying, then there you clean the back of the slide and mount to the microscope stage and examine under times 100 oil immersion. Then there, I'm going to show you when it is positive how it appears, and we differentiate with it when it is negative. So these are the steps we follow when we're diagnosing the TB. Don't forget to subscribe, because if you don't subscribe, you will miss a lot of work. So just subscribe and we stay in touch. Thank you for watching till the end.